Thank you. So my name is Jochen Kratsch. I'm from InCella GmbH, and formally announced was Dr. Gary Davidson to give this talk. So I had some problems with his journey, so I have the pleasure to introduce you the new generation of transfection reagents. Our transfection reagents called ScreenFect. And if we talk about cell transfection, we talk about delivery of 4 AIM genes into cells to have an output of protein, to have knockdown of genes, and many other applications. And there are many types of different cell lines, many types of different genes, so that there were developed a lot of different methods to make this gene delivery. There are methods like electroporation or microinjection, also virus-based methods, and what we are working with are liposomes. And in 2009 in Biomarket and Trends, it was said that transfection market claimers for silver bullet. So it says there is not one method which can do everything in transfection, but we are working on it to find one. And I will present you one possibility using cell-based screenings to find these silver bullet. First of all, I want to go to the mechanism of transfection, and we are using cationic liposome systems. And the positively charged liposomes are interacting with the negatively charged DNA, forming so-called lipoplexes. These lipoplexes can enter the cell using endocytosis, and after being in the cell as endosome, there are different possibilities how to escape from the endosome. First is the active escape from the endosome by mixing the membrane lipids with the lipids from the liposome, or by endosomal maturation. And then we have a lysosome with DNA fragmentation. So we want, in principle, to have this endosomal escape. And we can show, I will show you later, that we have this endosomal escape. And how important is gene delivery? Gene delivery is done in many biology laboratories. In fundamental research, in over 200,000 research labs worldwide, gene delivery is an important point. Also in 50,000 industry labs, gene delivery is performed routinely. It's also used in drug screening programs by biotech and pharma companies, and it's also important for large-scale production of pharmaceuticals. For example, antibodies, peptides, or proteins. But there are many unsolved problems at the moment with the current market products. Many of them have low efficiency, and high efficiency is especially important for large-scale transfection, because increasing efficiency leads to significantly increased yield in protein production. It saves time and reagent, and it can reduce cost, which is for protein production in large scale a big factor. Then a lot of current transfection reagents are highly toxic to the cells. There's a lack of versatility. There are many different cell lines, but there's no real multipurpose transfection reagent which can transfect all cell lines in the same quantity. There's a high price in transfection. A lot of transfection reagents are really expensive. Microinjection methods are really expensive, and there are also really diverse applications of transfection, so that makes it hard to have one good transfection reagent. So our technology is now based on lipids, on lipid chemicals, and these lipids consist of a hydrophobic tail and a cationic head, and this leads to spontaneously forming liposomes when mixing with an aqueous buffer media. And we use now combinatorial lipoid chemistry to develop many lipids, to screen many lipids in a short time to find really good transfection reagents for many applications. And We have three components. We have the hydrophobic tail, we have a linker and a cationic head. These are combined in a two-step chemical synthetic method, which is, in synthesis, is fast, efficient, and provides to high yields, which make it cost efficient, and we can offer our transfection reagents to prices less to our competitors on the transfection market. And here's our lipid library shown. We started our first library from 7 amines, 2 linkers, and 8 alkyl thiols, which were coupled in a two-step synthesis. First reactant is alkene with this alkyl thiol in a two-step chemistry reaction to this carbonylic acid, and in the second step we can couple this cationic head or this head group to the linker to obtain our final lipids. We have a total reaction times of 2 days. We don't need a workup of the reaction, and we can use extraction after the final synthesis step. We don't need to use highly expensive chromatography methods. It's easily scalable, and we can produce with this into a few weeks more than 100 unique lipid-like molecules. And this is how we tested our cell-based screening. We started with the parallel synthesis of novel lipid-like molecules. We started with 112. We extended our library now to over 200 lipid molecules we can use for a general screen. From this 112 lipid molecules, we can make 112 liposomal reagents by mixing the lipid with a helper lipid in an aqueous buffer system, and we spontaneously get these liposomes after 1 hour of stand. And then the third step how we use our cell-based screening is to test the transfection efficiency in different cell types with automated fluorescence microscopy. And doing this approach is shown here. So first of all, we dilute 50 nanograms of DNA in 8 microliter of our ScreenFect dilution buffer. We also dilute 0.3 microliter of the transfection reagent in the same amount of buffer, mixing them, and then you have to incubate this for 30 minutes at room temperature, and in this time the lipoplex is formed. After the 30 minutes, we add the cell suspension. Normally we start our screenings with HEC-293 cells and transfer this mixture to 96 voltage. These amounts given here are the amounts for one 96 voltage bulb. And after 1 day, we add Höchst staining and look for the fluorescent expression in our cells. And here are the results shown of our first transfection screen. We have GFP transfected 293 cells, which are shown, the GFP images are shown here. We compare this to lipofectamine as our internal standard and also as our biggest competitor. And we found that more than 10% of our new lipoids have higher efficiencies than lipofectamine 2000. And from this, about 20 lipids showing this high transfection efficiencies, we have chosen ScreenFect A, our first transfection reagent. Yeah. And our first library is published in Biomaterials. It was published in 2012, and it also was patented in the European Union, the US, and also in the many Asian countries. Now I want to introduce you now with a few examples to our first product we developed, what I've shown, ScreenFect A. ScreenFect A was launched in 2012 together with Vaco, which are distributing our reagents all over the world. And I want to show you a few examples. And the first example is here now shown in HEC-293 cells. As you can see, we can compete with the most important transfection reagents on the market. We have a higher GFP expression, we have a higher transfection efficiency, and we also have an optimal balance of toxicity and efficiency. So we have a really high efficiency with only a few apoptotic cells after 1 day incubation in the 96-cell bed. We can also use our ScreenFect A to transfect mouse embryonic stem cells. Here's shown chief image and the bright field image, and also you see here the both image merged. We have about 70 to 80% transfection efficiency when we transfect chief P with mouse embryonic stem cells. And many different other examples, for example, C2, C12 myoblast cells. Also here we have 80% efficiency and more. And we could also show that we are able to transfect very difficult to transfect cell lines. As here, zebrafish fibroblast PEC2 cells. The best on market known is Fueogene, and we can compete with Fueogene. We are also slightly better compared to Fueogene for this special cell line. It's also possible to apply the ScreenFect A in the biopharmaceutical industry, and I want to show you one example with miRNA transfection in rich media that performed in CHO cells. And we could show that we have a functional delivery of small double-strand RNA molecules in CHO cells. These pictures are now show the transfection of a fluorescence labeled miRNA compared to lipofectamine RNA IMX. So this is just show the transfection, not the expression of this. But we see that we can compete with a lot of other transfection reagents. ScreenFect A is shown here has the best transfection efficiency when you look at these pictures. It also have good efficiencies but high toxicities. This branched PEI polymer, this polyethylene imine polymer. But one big difference, and that's what I mentioned at the beginning, is this endosomal escape. We could show that for PPEI, the DNA is localized into the endosomes, wherein for ScreenFect A, they are localized in the whole cell. And this indicates that the ScreenFect A results in even distribution of mRNA throughout cells, indicates efficient endosomal escape, which is important for protein expression in the cells. We could also show that we have 96% transfected cells compared to 73% BPEI, and we are also able to have a functional knockdown in CHO cells. Here compared BPEI again to ScreenFect A. So this shows broad application possibilities for ScreenFect A. It shows also this is a really multi-purpose transfection reagent system. But it's not our only product. We have at the moment 2 different products on the transfection market. One is optimized for maximal siRNA delivery. ScreenFect siRNA, I will show you here some results. First of all, we've tested the LRP6 siRNA knockdown in mast cells, and we show that we are better than lipofectamine 2000, and we are comparable and slightly better compared to RNA IMAX. Also in CHO cells, we have here LGG production, stable CHO cell lines, and we see here that the knockdown with an anti-HER2 siRNA is also much better than to competitive products on the transfection market. ScreenFect siRNA can also be used for long-term gene silencing. This with lowest toxicity. Here are shown U87 glioblastoma cells, which are stably expressing chief P and luciferase reporter, and we could show that also after 7 days after transfection, we have nearly total knockdown of the luciferase in these cell lines, transfecting this acyl luciferase RNA with ScreenFect siRNA. The last and our newest product, just on the market for a few weeks, is ScreenFect mRNA. it's an efficient and general reagent for introducing mRNA into cells. And once again, we used our lipid library to screen many of different liposomes, and we've chosen here now 6 which are showing transfection efficiencies of 90% and more. And we choose from this ScreenFect mRNA showing transfection efficiencies up to 100%. And here's also shown comparison to mRNA with cells with no reagent, so we can provide you an mRNA transfection reagent with efficiencies up to 100% shown in HeLa cells. And these ScreenFect mRNA can also use for reprogramming stem cells. And here again show the comparison of ScreenFect mRNA with lanfectamine 2000. And as you see, we have a much higher expression in GFP, so we have much higher GFP intensity, we have less toxicity, and a higher total efficiency. And I've shown you now 3 of our current products, but we are developing more and more products, and there's very different applications for transfection reagents, and when we try to cover as many as possible of these, and we're working on many topics, especially for reagents for large-scale transfection, also for in vivo gene delivery. We are working with different cooperation partners. And currently in development is ScreenFect A, a transfection reagent specially designed for large-scale transfection, ScreenFect IPS for transfection into iPS stem cell, and ScreenFect MEF, a reagent which should be suitable for MEF transfection. MEF cells are also hardly to transfect, and we want to provide one specialized reagent to transfect mouse embryonic fibroblasts. Thank you for your kind attention. And if you have interest in testing our samples, please feel free to contact Marco to provide you some test samples. And thank you again for your attention.